Hi guys. We're continuing with life science in a nutshell. We're going over my notes. Um, we did. This is the seventh part of this science section or review. We have reached the section of we're doing cell membranes and okay, so we're learning about how a cell's external environment affects its shape and also how molecules move across concentration gradients across the cell membranes, depending on gradients. So there's membrane proteins that help with this kind of molecular transport or movement, especially for larger molecules and polar molecules like sugars, amino acids, etc. You need help in the form of membrane embedded transport proteins, which help this kind of movement. Um, so, and when it's against concentration gradients, it uses up the cell's energy. However, down gradients, it doesn't use much energy. Ruben's gonna help me read notes. Say hi, Ruben.
Hello.
So hope you can hear. We're gonna try to be a little bit louder. Sometimes it's not that easy to hear. Okay, microtubules. So Ruben, you want to read that?
Microtubules in interface cells, plasmosis,
Osmolysis.
which is the disruption of the cell wall plasma membrane, cortical, cytoskeleton,
Continuum.
continuum is expected to exert the strongest impact on cortical microtubules since they are closely linked to the plasma membrane, exerting a role in oriented cellulose microfibril deposition. At the onset of plasmolysis, cortical microtubules became wavy in order to accommodate the decreased shape of the protoplast.
Sorry.
Concurrently, the microtubules come together to form. Who cares?
So they form bundles. Go ahead.
But a row of microtubules in the transport of cellulose synthase, uh, CESA proteins from their origin in the Golgi towards the plasma membrane. And
hold on one sec. Okay, so plasmolysis causes the membrane to pull away from the rest of the cell, which has the cytoskeleton. So so the microtubules, which are part of the cytoskeleton, are changed in their shape, and so go ahead.
in this plasmosis, microtubules remain organized in thick bundles, but they are gradually separated from each other during the course of the protoplast, swelling to its original size.
Okay.
That'd be a plant, right? No proteins.
So here, let's see, swelling. So once you have deep plasmolysis, which is the opposite of plasmolysis, plasmolysis is water leaving the cell. Deplasmolysis is water entering the cell. So you have the microtubules going back to their regular shapes. So they're less spongular, I guess you could say, because the water makes them more swollen. And this is showing some of the stained cells of plants and the microtubules and how they look like with plasmolysis and deplasmolysis. So these are from experiments. And then another part of the cell that the research studied in plants as far as plasmolysis goes, they're called microfilaments. Actin. You want to read that?
Actin microfilaments, filamentous actin as visualized by GFP, ABD, and Arabidopsis.
Yeah, that's the species of the plant that the research was studying, like these cells shown. Okay, go ahead.
Hypocolloidal cells presented a fine network. It consisted of radiating microfilaments and microfilament bundles, which extended from the nuclear envelope towards the cortical parts of the cell. Hyperosmotic stress induced actin filament reorganization in leaf cells of Chlorophyton chromosome J.
That's just the research, okay?
Example by 2002. Filamentous actin as visualized by GFP.
Presented a fine network. So the plant cytoskeleton controls the regulatory volume increase and decrease. A lot of current knowledge on the water balance of plants is based on extensive studies by Stadelman and his coworkers. In his study, although the focus was maintained on the cytoskeleton, though part of a complex cytoskeleton plasma membrane cell wall continuum, plays an essential role in signaling and mechanosensing. In leaf cells of Chlorophytum chromosome, the behavior of cortical actin filaments during plasmolysis has been described in terms of plasma membrane integrity and protoplast volume regulation. In this study, actin microfilaments were shown to line the plasma membrane at areas of intense mechanical strain and, as in the case of concave plasmolysis, the plant cytoplasm. So we saw that earlier in the diagram. Where'd it go? So these are all the membrane proteins we saw earlier. Right here in this little diagram is showing the way the cell membrane and the inside part of the cell kind of separate, and the space grows between them as the water decreases, and this causes plants to lose water pressure and to wilt. So that so the protoplast volume regulation in this study, so the plasmolysis occurs, and then the plant cytoplasm contains a pool of globular actin in order to constantly assemble filaments. These in turn rapidly disassemble if actin remodeling is necessary. Actin microfilaments reorganization after hyperosmotic treatment. So when the water is increased on the external environment, was linked to calcium signaling and was associated with membrane mechanical integrity and references, etc. So we're going to continue in the next video. Thank you guys. Bye.